thing when we talk about genomic to sequence a genome as well. Then we want to talk about how on earth do we interpret it? How do we know whether it's making a difference in a human and making a medical difference for people? So let me start with what a genome looks like. And if my slides are going to move. So some of you might be very, very familiar with some of these images, and some of you might not. But this one here, okay, so this is an image of a genome. It is the whole genome. It is a microscopic photograph of a genome. And as you can see, you'll probably be able to pick out immediately, well, these are clearly chromosomes. Now, chromosomes, what's interesting for me, I always love where words come from. Chromosomes are staining bodies. So that is what a chromosome means. It means that you can stick a stain on it. And if you can see it under the microscope, it's a staining body. So it's a chromosome. So these are chromosomes. This is the entire human genome on the slide. And you can see there are 2 red dots there. Now, the point is, of course, that there are 2. And that is because actually, this is chromosome 6 that has been highlighted here. But the point is, with them being 2, we know that we get chromosomes from mum, we get chromosomes from dad in terms of biological reproduction. There's an equal contribution in terms of pairs of all of the chromosomes, and that's what you see. So we see chromosomes, we see that there's pairs of chromosomes, and if you know how to identify chromosomes, you can pick out the different chromosomes on that slide. Now, this is also the interesting thing here. So you go from this, you can't see the DNA yet. Okay, so we're seeing the chromosomes, but we can't see the DNA yet. So this is where we start to go into a little bit of a higher magnification. So this is electron microscopy, and this is one of the highest magnifications that you can get. So here you start to see these strings of beads, and that's what happens when these chromosomes have been teased apart. Basically, you sprinkle a little bit of salt on it for want of kind of more sophisticated kind of descriptions, but it's essentially a little bit of salt, and it starts to essentially spread out and pull out these strings of beads. Now, if you go into higher resolution still, you can see on the left-hand side, there's like a skeleton out of which are coming these enormous strings. And that's the DNA. So that's the DNA that is packed into every single chromosome. And that's the DNA that essentially makes up the genome. And that is what really interests us when we're talking about genomics. It's the DNA that interests us. Now, really at this point, we've got to the limit of microscopic resolution. So everything else that we know has been built around models. So like I was saying, the model that Watson and Crick built of DNA. So this is the very famous double helical structure that they discovered and that they announced that day in the pub. And it's also built from imaging. Or you could say it's someone's imagination. But these images, we know very well that actually what you can see is that there's a clear structure. When you tease this apart even more, there's a clear structure to those long strings of DNA. And that model, as we said, was found in Cambridge. And what was important with what they discovered there was this ladder type. So you can see when you're looking at this, there's a ladder type model going up. And then you can see that on the end, there's each rung of those ladders is generally known to be, or it's called a piece, or it's a byte, actually, BYTE. And that's one byte of genetic information. Now, chemically, you'll probably know this, be really familiar with this. What is encoded in all of those atoms are carbons, hydrogen, bit of phosphorus, all of those sorts of things. And what's encoded in those steps of the ladder in each base is either an A or a C or a T or a G. And that is essentially the 4-letter alphabet that we talk about that makes up DNA and makes up all the information for each of us that has become known as our genome. And so the nature of what we're trying to do is really unpack that letter alphabet, to understand why it's in particular order, to understand if it gets out of order in some way, what does it do? What's the impact of this? And what's happened therefore is we've come to know this as sequencing. So we've come to know that sequence of DNA of the letters as being a sequence, and therefore a sequence of letters. And hence that's why we kind of talk about sequencing. And we use the analogy of a book. That's not necessarily useful in many cultures. In Aboriginal Australia, people like Gareth Bainham have done incredible work with life languages that have taken out the song lines of Aboriginal culture to start to explain what DNA looks like, and what that can look like in terms of dams or blockages, or how we start to read that story and that pathway. But what people are trying to do is to understand the structure of letters, the diversity within it that expresses human health, also human disease, and all the aspects that might help us to really understand where can we pinpoint treatments even, or move diagnosis forward. So what's in a genome? Very classically, 46 chromosomes in pairs, 3 billion bases, again, times 2. We know that there's more than 22,000 genes. There's a lot of discussion around how many genes exactly, and there's mutations which cause disease. So once we got to the end of that, of course, these are some more facts and figures about DNA. So what we've just explained, you've seen the double helix structure there. You can see the sugar phosphate background, which helps to build the ladder. And then you see the A, the G, the C, and the T. And generally these nucleotides, so they will always bind generally together. And as we've said, 46 chromosomes, 22 plus an X or a Y, which generally denotes sex in some way. They talk about this being 2 meters in length. One of the descriptions I really, really love when we're talking about DNA is imagining it like a ball of wool, all rolled up. And then what we've got to do to be able to read it is unroll that ball of wool and start to kind of piece it together in terms of, okay, what's the order, and read that sequence as it goes through. So I do just want to talk a little bit about what happens in sequencing. So, sequencing, I think for me, it has, when I first heard about it and first worked with teams in Cambridge about it, there was kind of this idea that it was a box and something magic. There was library prep, something called library prep that happened beforehand, which then I came to understand, having gone through the cycle of taking my own sample through a mouth swab and then going through all the procedures, the recipe building to extract the DNA from that, then doing everything that I needed to do to that DNA to be able to put it on a sequence so that a sequencer could read that DNA and give a result. Just want to explain a little bit about what happens in the machine. So essentially, in sequencing technology with Illumina, there's short reads. There's also long read technology that PacBio, Oxford Nanopore generally are using. And the long read technology tries to keep the whole strand of DNA whole as it strives through. What we do, sometimes that means that there's misses. Sometimes that means that accuracy is not necessarily as high as it could be. What we have been trying to do at Illumina is we take roll the ball of wool out, and then we cut it up into lots of different pieces, and we put a tag on it, and then we put it in the machine. The machine reads it loads and loads of different times, and then in reading it, it produces these images. So every time you've got the G, the T, the A, the G, the C flag up, its corresponding partner should flash up at the same time. And so you see all these images which are colorful. Essentially, it's lots and lots and lots of photos that happen in the machine to then be able to roll this out into what you call reads. So like you're seeing here, CGA, CGA, CGA. And they do that a number of different times. So that's what we call depth. And then they base it against a reference genome. Okay, so there's a lot of discussion about reference genomes. Obviously, the Human Genome Project was the first real reference genome that we had. And often, we're very critical of that, and that it seemed to be a white European male. And actually, many people even give that white European male a specific name as to who that was. But essentially, now we're building up reference genomes depending on location, geography, depending on populations, depending on disease areas as well. And, we've already referenced the 100,000 Genome Project. Part of that was thinking about how do we develop a reference genome as well. And then there's interpretation. Now, interpretation, we can talk about this a bit later, because when we're talking about access for children, families, patients that we know and in our groups that we represent, we often know that this is a stumbling block still. And I think the important thing here is to remember that we're at a particular moment in history with this. So while there has been a really, really long journey in terms of the technology being able to do this piece, so to go from Watson and Crick's discovery and thinking about the micro electronscopy, the resolution that we can get on all of this and how we move forward, we've still got a lot of work to do to think about how do we interpret the information that's coming off. So how do we interpret the sequence of letters and make decisions about, is this impacting health? Is this not? Are there going to be possible treatments that we can, access or target to these specific areas? And what does that mean? And there's a lot of work being done around making sure that analysis becomes a little bit more consistent, that there's quality to that we align not only hospital to hospital or lab to lab in terms of the results and how we interpret that, but country to country and region to region. But I do want to say at this point, and this is one of the most frustrating points for me at this point, and this is where I'll talk about one of the stumbling blocks here, is we've still got a long way to go in terms of understanding this. The more whole genome work that we do, the more what we call VUSs, so variants of unknown significance, we're finding. We need to do a lot more work to understand what is happening there. But this is just a simple slide to think about, what can the impact of an error as an error in the genetic code actually be? And some of these are very simple. So a G to a T causes a major change in a protein. And it depends on, it can be a single letter difference. It can also be that there's a relocation, so it can be a deletion, it can be a duplication. It can be that a whole chunk of the sequence is completely moved to another place. So we can have translocations, we can have all sorts of things happening. Or here you can see that where a G goes to a T in another area, it can cause an actual termination of the protein. So there's a whole lot of different things that can happen when one letter goes out of place, or when there's a transfer of a G to a T in terms of what impact that has. And it can be that the impact is downstream, it can be that it's upstream, and how these regulate in terms of understanding the mechanisms of biological causation. There's a lot of work to still be done here. Having said that, I do just want to flag up that actually, again, this moment of history that we're in, we still, I've been in Brussels this week. Last week I was in Greece. The week before I was in Poland. And each week, the question has been from advocates and clinicians alike is like, we still need to bring the cost down. So there has been a huge reduction in cost. We estimate that the Human Genome Project, or the first human genome sequencing, took 10 years. It actually took 10 years to achieve one genome at a price there that you can see. And then we're moving this down at the moment. I mean, this is even a little bit of, this is really out of date, actually. We're moving this down and down and down in terms of actually we're getting it down to around 300 at the moment. And we can turn it around depending on the genome being sequenced within a 24-hour period. So even less in the case of the rapid whole genomes that are being done. But this is something, from a patient advocacy perspective, I'm going to be really upfront in terms of we want to see this cost come down more and more and more in terms of making it applicable and accessible. What I do just want to say quickly as well here, recognizing that I want to leave plenty of time for talk is for discussion, is that one of the things that does happen often is, and we can talk about this in in terms of what your experiences have been, is there's a lot of talk of different genetic testing. So often what happens is we have broken these down into particular genes that we know of that are going to be responsible disease causing in specific examples. So for example, if it looks like you've got lung cancer but have never smoked, then it's highly likely you might have an ALK positive test first, because that can be turned around really quickly. So they'll test immediately to see, is your tumor, not you, but is your tumor ALK positive? Then if they get a no for that, they might do a broader panel, which includes, EGFR or a couple of other known genes in that space. And then again, if that doesn't happen, or if there is a sense that there's been cancer in the family or other cases within the family, you might want to look at a whole genome. And in the case of cancer, it would compare the tumor, so the somatic cancer cells, so the changes that happen in the actual tumor, with the germline being your actual genome and your germline, what we call germline. So what we're finding more and more is that, again, this moment of history that we're in, that I often have in the rare disease space, two families come up to me and go, well, Shirlene, we've had genetic testing and we haven't found anything. And then you end up pushing and finding that actually they had different forms of testing. So they might have had a FISH, they might have had what's known as fish or Sanger sequencing, or they might have had a panel, because it was assumed that epilepsy was the strongest symptom, and so they've had an epilepsy panel test. And increasingly what we're finding is that we need to do more and more whole genome or whole transcriptome sequencing to develop the most comprehensive profile in terms of its impact for really understanding the broadest possible applications how we can translate that. And that ranges from everything to do with what you would assume, say, in cancer. This is particularly looking at cancer from hereditary cancer risk right through to metastatic, or at diagnosis, then how do we monitor residual disease, or how do we monitor disease progression as well as people go through treatment? So there's kind of a long potential of where this technology can apply. We haven't realized that at all in any way, shape, or form. In many cases, we know in certain patient groups that, of course, this is going to be of value, but it's really, really hard still fighting for reimbursement or fighting for access. And, again, we'll talk about that soon. But the really interesting thing for me, is, and I won't show you the actual genomes now, but is when we're looking at signatures too. So, for example, if we're looking at melanoma or lung cancer caused by smoking, what absolutely blows me away is that when we do the genomic analysis and do a whole genome, you can actually see the signatures of those moments of environmental exposure. So you can see the changes that have occurred in DNA at those moments of either sun exposure or smoking exposure. And we've got a lot more work to do to uncover that going forward. But this is a little bit provocative, but again, this is something that we play with, and certainly with the number of advocates that we work closely with, we've kind of talked about this. This is today where generally we think about this. All these little arrows down and arrows up are not what we're suggesting here should happen or even do currently happen, but they're where it's kind of thought, okay. So you'll know at the moment in the UK, there's a whole lot of discussions around the generation study where newborns are being sequenced at birth as a kind of genomic newborn screening research study to explore the impact. So there's a lot of people who talk about, oh, if we could just have a genome at birth, then we'd know everything we need to know. But what we're trying to show here is that that's not necessarily the case. It's not one test once, know everything, because of course our DNA, there's what we inherit, but there's also the fact that our DNA changes according to exposure at different points in time. And the DNA of cancers, the DNA of diseases can be very, very different. But this is just a kind of idea of, okay, a schematic. What would it look like if we did more testing? So what would it look like if, you know, we take for granted that when we go in, we're you know, you're pregnant, you automatically have a scan. In fact, you know, increasingly people are asking for more and more scans through pregnancy, or you go in, suspect a broken arm, you have an X-ray. What would it look like to have genomics as a backbone in terms of our interpretation of health and our understanding of health? And when would it make sense? And what sort of tests at what point would make sense? So whole genomes at what point, panel tests at what point, you know, little tests to kind of check on relapse or disease metastases in any way, those sorts of things. So this is something we can discuss later on. But we do strongly believe, and I as a sociologist, I've been a bit cynical about this in the past, but I do increasingly and, you know, very strongly believe that genomes actually will be transformative to the practice of medicine. At the moment, it's incredibly disruptive. It's not commonplace. It's not how we're trained to really think about disease in many ways. And we're still struggling with that interpretation piece that I talked about earlier, that analysis piece. But the potential here is that genomes can give us really precise diagnoses. So we know in many cases, and you'll know this from your work, that we see misdiagnoses again and again and again, and misdiagnoses often impact on an ability to get to treatment on time, you know, in a timely manner. And so that will change. We've got earlier detection, the possibility of that. We've got interventional prevention, but there's really strong hope here that this could impact across all these areas. And just before we go into the discussion, I wanted to give you a couple of examples of things that maybe, you know, stand out in ways that we work together, or, you know, the questions that we often have in this space. So if I give you this one, you'll be aware of the Tessagel Brain Cancer Mission in the UK. And Nikki, you know, she has remarkable access to really think about what is happening in this space. So in brain cancer in the UK, whole genome transcriptome sequencing is fully reimbursed. So every brain cancer patient in this country should get access to whole genome transcriptome sequencing. And Nikki, in one of her analyses, her team found actually in this country, only 5% of brain cancer patients were having whole genome analysis of any sort. And she was just like, why is this the case? And so she did this piece of work, which ended with Closing the Gap, which came out in September 2024. There's been another piece that has come out a year on from that, so last year, end of last year. But one of the things that I just want to highlight from the Closing the Gap piece is that she really delved down on the pathway in England. So you'll see the diagram there to really think, okay, what happens? And, you know, there's several pieces by doing this. She has a kind of wonderful series of slides of unpacking this pathway and then starting to show where the barriers are, because, you know, you and I probably all are very, very aware that sometimes it's like, oh, if only patients would ask for that, they'd get it. But we know that there are systemic barriers to access. And she was able to identify very fundamental systemic barriers. When you've got your needs to consent for the tumor tissue that needs to be stored fresh frozen. That's one consent, that's one process. And then you need the germline consent. So you need the blood test result, which is a blood test consent, which is also another practitioner. So you've got two different samples that need to come together, two different consents. Often she was finding that people didn't even move beyond that. So you might even never get past that first initial consent phase in terms of a doctor being willing to take a germline consent or the surgeon actually storing the tissue appropriately. If they did, those samples need to come together in order to be transported to a plating hub. And it was no one's responsibility within the NHS to order that transport. So no one did. So often, again, you saw a huge fall off there. And then again, coming back to analysis and moving this through the Genomic Tumor Advisory Board and giving information post-analysis that was appropriate was, again, another huge systemic barrier. So these are just, examples of that's one example of a piece of work around the pathway to really analyze this, and now to go through steps of, okay, we know where to intervene. What do we need to do? How do we give a why? How do we make sure that this part's streamlined? Has been really powerful. The other example is in rare disease, and this is, perhaps an unusual example, but Alison Watson, who is the lead of the Ring20 Research and Support Organisation in the UK, it was 7 years ago actually, and she presented at the Sanger Centre, saying that since we have had the impact of next generation sequencing, we are seeing a downturn in our diagnostic rate for our community. And the question then was why? Basically, you can see on this axis at the top, where it's kind of we've actually mapped diagnoses. These are globally, and we again talk about the misdiagnosis and the undiagnosed within the Ring20 community as well. But the reason being that, as I showed you right at the beginning, the way that Ring20 is diagnosed is actually a karyotype testing. So looking down the microscope, counting out the chromosomes and seeing that there's the 20th chromosome and it's formed a ring. And that's how a diagnosis occurs. But what's happened is that we have then, as we've introduced exome or whole genome sequencing, the ring cannot be observed at the moment. Deletions might be detected, but we cannot observe the ring through genome sequencing. And there's a number of reasons why, but what we've realized, of course, is that the ring is not the cause of this condition. It is a symptom of something else, and we need to find that biological mechanism that's causing that. So we developed a program with that, where we developed what we called the Unravel project. This has been a major education piece. We created assets like you see on the screen here, to really start to educate the community about, well, I've got a diagnosis, why is this going to matter? And the reason it matters is because we cannot develop cell lines, we cannot develop animal models, we cannot develop any research to think about treatment targets for this condition at the moment. So it is all completely experimental as to how we might treat these children who are diagnosed, and these adults who are diagnosed with Ring20, to improve outcomes. And then what we wanted to do was move to a study where we collect samples from people with the diagnosis of Ring20 to start to see if we can really throw the book at it in terms of trying to read that genome and find the actual cause. What is the change that is causing the ring to form? We're not there yet. It's been a 7-year project and we're still going. But and that's what I was saying earlier about this being a moment in history in terms of there's what we can do at the moment, what we can analyze, what we can interpret, and there's also still a whole lot more that we need to do. But this is an example of another project that we have worked on together. So I can talk a little bit more about all of that, but for now, I think I'll leave and stop sharing, and then maybe we can have a discussion, because I'm very aware that we're already, and you can ask any questions.
That was brilliant. Thank you so much, Shirlene. So we'll hand over to the floor then for any questions, if anyone's got any. I mean, I could start just with a sort of an obvious one, I suppose. I mean, how could we get involved as charities, and why should we get involved in this, given that it is very early stage? And as you say, there is, and I suppose that the question next to that, I suppose, is the $1 million question, which is, do we want to potentially know that we've got something coming down the line to us if we know that there's no treatment?
Yeah, that's a really interesting question. I'm going to start with that one because actually I talked about that yesterday in a presentation in Brussels. So, so in the rare disease, I think it's useful to think about the rare disease space here, and Jane's very familiar with this. But if we think about the rare disease space, so those people who had a diagnosis, often that had happened through particular research or through clinical investigation. So for a clear phenotype that you were able to diagnose that on. What then happened is for those, those few conditions, we had a number of different moments that unified the patient organizations that were around kind of the creation of what we call orphan drugs and access to those orphan drugs. And so what happened in the rare disease space was diagnosis kind of got forgotten. And really it wasn't something that we focused on a lot in the pathway. And as we've introduced whole genome, as we've identified, so what's interesting is the number of rare diseases that we know exist globally is rising all the time. And partly that's because of these new technologies that are able to identify them. So we still estimate that there's about 60% of the world living with undiagnosed rare disease. So these are people who still cannot get a diagnosis, even using the best possible technologies that we have. So whole genome sequencing at the moment is seen as the most is the highest in terms of diagnostic yield, but we're still only, even with the best sequencing, getting around 40 to 50% of a diagnosis through. So what we know also is that actually there's a lot of people who have been diagnosed, but I had a pediatrician, for example, say to me one day, well, we know what's wrong with that family, but there's no point in telling them because there's no treatment. And the importance there is, and again, this challenges me as a sociologist, because as a sociologist, historically, we kind of learned that diagnosis can, in some cases, create stigma. But what we also know is that diagnosis creates the possibility of building an ecosystem. So if you have a diagnosis and then you can find someone else, particularly in the rare disease space, even if that other person is in Sweden or in Australia, you can start to build some sort of community, and you can start to create some sort of ecosystem by which you then start to build the research, you start to build the innovation in terms of even building therapeutic options. And so what we are seeing in the rare disease space, for example, is we're seeing a real kind of reclaiming of therapeutic development by patient organizations. So in order to get things through, they are developing whole new platforms by which we are starting to look at developing therapies. So some of you will have heard of the N=1, the plausible mechanism pathway that has really kind of come through in the last couple of years in terms of, okay, if we've got this specific piece of information and we know that we can target it with this particular method, can we apply that to a whole lot of different diagnoses as well? So the mechanism actually becomes something that you get through the FDA, that you get through all the approvals, but the target might be slightly different. And so we have what's called N=1 therapies emerging. So what I'm just trying to say there is that often, we, again, recognizing the space that we're at, diagnosis is incredibly important in and of itself. But it's not the end. It's not kind of like long diagnostic odyssey, get to whole genome, hallelujah, that end. It's like it's actually about what work does diagnosis do for creating new avenues for research, new avenues for how we understand healthcare, new avenues for therapeutic intervention and its applications, and the way that we do that. And sometimes, again, those are really specific. So for in the case of epidermolysis bullosa, we, you know that that's a rare skin condition, that's quite highly visible, but in sequencing we've been able to identify the subtypes, and the importance of knowing those subtypes means that it can be little things. So there is a gene therapy, there are other things, but it can also be as simple as we know how to dress the digits, how to appropriately dress the digits of children so that they won't end up being amputated, because that subtype, there will be webbing, this will happen. And that's what we need to understand what we're doing here, so that we're not guessing and experimenting really in terms of hoping, oh, this might work, this might not. If we know with precision what the cause is or what subtypes people have, then we can start to even, think about those sorts of things. So that's the first, why should we pursue this? I think, again, I think we're at a really, it's difficult because as advocates, we're always taking everyone by the hand and showing them the future that we want and trying to get there. but what can we do to be working together? So, at Illumina, I will just say we don't have the resources that pharma has, where it's slightly different in the way that we operate. But, my team, Sergio's on here, we are very, very keen to partner. And one of the things that Illumina does have as an extraordinary resource is incredible thought leadership. So, sometimes we can partner on research that very much is like the Ring 20 project has not been huge investments of money in any way, but it's been huge investments in terms of time, in terms of really, what are we learning about Ring 20? What do we need to know going forward? How can we move the space? How can we kind of analyze what material we have and build research? We can also do education programs. We can do a whole lot of different things that we can support you with. We have the foundation as well, which does support independent initiatives. And yeah, there's a number of different ways that we can get involved where it makes sense for your community. I see two hands. Melissa, I think yours was first.
Hi, yeah, that was really great. I really love the detail. My own kind of, in my previous life as a researcher, my PhD was on RNA sequencing, so super familiar with the technical details.
Okay,
But now that
sorry.
I've crossed over, no, it was good to hear kind of the updates since then, because that was ages ago. But now that I've crossed over onto kind of working with patient organizations, I'm almost at the other end of kind of the problem. So, just for context, I work with patients with hearing loss and tinnitus. There are currently no
Yeah,
therapies beyond devices for
no.
them. As of this year, we have the first genetic therapy for our ultra rare form of deafness. It's amazing and it's completely restoring hearing in these babies. So, our current problem is super basic, but it's communicating what, like things like we're getting questions, what is a gene? What is DNA? What is a
Mm,
genetic test? And that's what we need to be answering and providing that really reliable information to our communities at that super basic level that's
yeah,
independent of the pharma companies that are producing these products. So
yeah,
we're doing it on an ad hoc basis, but we would love to create an information piece about how do I access testing, what this is, things like that. Do Illumina work in that space? Because obviously I am aware that you don't make therapies, but do you have that kind of lay aspect to the work?
yeah. So we've worked with a number of different organizations to create fact sheets or information on those really basic levels. And that's what I was saying. We can partner very much in terms of thought leadership. So we have, a huge breadth of team that can, breadth, of experts who can also check different pieces around terminology. Even how do we explain a gene? How do we make sure that things are accurate in what we're describing? um we've got we could connect with you offline. Sergio can also connect. We can connect because there are a number of groups that we have worked with who have done this, obviously through a number of different programs. What's interesting at the moment is historically we didn't have our own Illumina patient advocacy facing page that might have some of this information, because it was kind of
Mhm.
seen from a compliance perspective. And as you were saying, keeping that information neutral, it's
Mhm.
a fine line. But again, it's not like pharma. So we are being asked more and more
Mm.
by patient organizations, Shirlene, can you just do that and have somewhere that we can all go to rather than reinventing the wheel all the time with every single
Yeah.
organization? So that is something, let's discuss, but that is something that we can really help you to do in terms of fact sheets, or even just point you, look, actually, we think that's a really good one. They've developed that for that. It might be
Mm.
a different, but maybe you can adapt that for you, and we could, help get, either ourselves or also bring some other teammates in to just, fact check it a little or sense check it a
Mhm.
little, what this
Yeah.
means, but can definitely help with that.
I mean,
Great.
I think
Perfect. Thank
that's
you.
something that probably a lot of charities would want. So maybe it is something that we could offer out to different organisations and sort of have something, because really it doesn't matter, does it, what disease area you're in? You're asking exactly the same questions. Yeah, what does it mean
Question.
to us? How do you get it? So
Well,
yeah, maybe
and so
it'd be
like
a nice
even
STAR initiative.
yeah, because even with the Ring 21, it was really important, because it was kind of like, well, why, you know, we've already got my diagnosis, so why do I need to know this as well? So it's kind of, it wasn't even people pushing for a diagnosis, but it's kind of those people who had a diagnosis even understanding the diagnosis they had. So we created a karyotype kind of I can't remember, demystifier tool. So you could put in the actual thing that came back on your report and have each letter and number explained to you in a whole lot of different ways. We created animations as well, all sorts of things. There's loads that we can do in terms of thinking about how do we communicate this information. Yeah. Jane.
I've been around the block quite a bit, and I started out thinking, well, genetics and genomics only has to do with rare diseases and inborn errors of metabolism. And the older I get, the more I realize that at some level and to some degree, every disease and every condition just reduces to some element of the genome and the environment. And in terms of translational research, researchers and innovators, as part of developing their products, are generating unprecedented volumes of genomic data through translational research. So I wondered if you could reflect on what strategic shifts are really needed across infrastructure analysis and clinical integration to truly sort of capture and maximize the impact of all this incredible data on patient outcomes. Hard question, hard exam question for you.
Yeah, at 12:56. Thanks, Jane. I think you touched on a really important point. And so, for example, in just to give you an example of something that, you know, we have done to respond to this, is that often what we've seen, particularly in the cancer space, is that there have been pockets of developments of information. So, for example, and they've always been tumor specific. So everything has operated within some sort of tumor specificity. And suddenly we're finding that, you know, EGFR positive is not just lung cancer, it's also in brain. I mean, I had a talk yesterday actually where it was also an overgrowth syndrome, and, you know, where it is in the pathway. So, you know, the thing is that we're starting to uncover that these variants that we're finding in tumors, let alone in the germline in tumors, are not tumor specific in the way that we have categorized cancer historically. And so that's what I was saying earlier, that this is really, really disruptive. And it's like, how do we start to use that information more appropriately? We're starting to see it outside of the UK in the DRUP-like clinical trials. So we are starting to see, okay, let's do an analysis of the tumor first up, you know, right at the beginning of diagnosis. And then let's look across the tumor to put people into trials, so DRUP-like trials based on the mutation rather than their tumor type, and giving people access across countries that they probably haven't had before. So, you know, an esophageal stage 4 cancer patient finding out that they've got a HER2 mutation, which we normally associate with breast cancer, then being able to go on to a HER2 therapy, which is only really fully reimbursed in their country for breast cancer, but actually getting to go on a trial because of that. And, you know, it proving that it worked for him. It actually, within 3 doses, reduced his tumors, and therefore then putting the responsibility back on pharma that actually they have to provide these therapies for people across region, even if it hasn't been reimbursed in every single country. So this is not answering your question specifically, but what I'm saying is that I think as advocacy organizations, we have a big responsibility in terms of that translational piece of really showing that we can't be siloed out anymore, that for this space to really have relevance for us going forward, we need to be working together and we need to be working across our groups. The big piece that you said is data, and the big piece is analyzing that data and thinking about how do we share that data, how do we make sure that data works for patients so that it moves forward. There's a lot of different programs that are starting to move in that space, but I think it's one of the biggest barriers that we have at the moment. It's still a big barrier across Europe, let alone then where the UK sits in terms of data sharing. It's still a big barrier in terms of being able to move to other places to access therapies that might not be, available in your country, even though the data might suggest in another country that this is highly relevant. So, I don't have a clear answer on this. I think there's increasing attempts to do this. There's programs like the Genome of Europe that are thinking about how do we create reference genomes at a population level rather than at a disease level that can then help us to think about this information. But translationally, Jane, we're right at that bleeding edge of it at the moment in terms of the how, the what, the way forward. I think it's not clear. And I think we've got to develop the pilots that show, that exemplify the why. That was the big piece with the Tessa Jowell brain cancer mission, actually, was then thinking about, we've really got to exemplify the why here in order to then make it more obvious as to how we might translate that data. But Sergio, you might have some things to say about that because you're my data man. But I don't know. I think there's a lot of work to do. Short answer, a lot of work. How? I'm not exactly, I don't have I think we should do it this way answer just yet.